Okay, let me invite our speakers, all four speakers, to the table so we can have more questions and discussion. I think this has been a fascinating session, and I guess for an epidemiologist like myself, the parallels between the symbiotic relationships across the board, from plants to insects to higher animals, is just very, very striking. The importance of the equilibrium. I think you all four in different ways touched on the importance of that equilibrium between the host and the microbes, and how the perturbation can have deleterious effects, is very striking. I see people come up. I'm actually filling time here until people think of questions and get started. But actually, just to ask, can you comment on across-the-board parallels that you might have noticed from each other's talk on that, the importance of the equilibrium and how disturbing it can really send us astray? Any one of you?
I think one of the really important issues that we need to address, and perhaps will come up in some of the later talks, is how robust particular states are. And I think this came up in several cases, that both healthy and unhealthy states can be highly robust to perturbation. But once a perturbation arises, then they can shift to another stable state. And I think as we look forward to future talks, this may come through as a general theme.
Yeah, I mean, you know that enterotypes in the gut have been described. That different people have, at least the French studies suggest that you have kind of three types of bacteria communities in the gut. This is controversial, but it means that you have different states. We don't know the impact of these different states on the state of the immune state, or the protection state, or the disease state of the host. But that goes back to what you said.
Okay, Jackie?
Yeah, I wanted to go back to Dr. Tuscan about your talk about the populace. And I noticed that you focused on bacteria and fungi. And I wanted to ask, well, I suspect that you just kind of put oomycetes into fungi, but you didn't mention viruses, and that was brought up later by some of the other speakers, and of course they're there. They would have a direct effect on the plant, obvious effects on the microbes, and a source of genetic information. So how do they balance into your equations?
They're clearly there, and the amount of diversity that we see is almost overwhelming in a sense, and trying to partition that into those that have intimate relationships with the plant and those that are just associated by coincidence, and those that are just present for other purposes is very difficult. And so we don't have anybody specifically looking at it from the plant perspective, but we do from the bacterial perspective. I think it's an area that merits further research. We know many viruses are pathogenic, that cause many perturbations in plant function, but it's not an area that we're spending a lot of attention on, but probably merits it.
Just also to you, I'm just thinking of some of the decimation that's happened to some of our great trees, like the chestnut and the elm. And you made a very strong point about the host being the driver of what is the community. I wonder if there's any role for other members of the community, the microbiota, in tempering some of these diseases that have taken out whole species.
In most of those cases, they're exotic pests or pathogens, bacteria that have come into the ecosystem that the host wasn't confronted with during evolutionary time. We do know that inoculation of sterile poplar plants with a suite of bacteria can convey acquired resistance to other pathogens. It's likely that it is possible to find a microbiome in the native environment where these organisms are present and are not pathogenic that may be beneficial to the host in this continent or in other situations. I think it's a very good hypothesis.
Jesse?
You know, it's really a lot of, I remember this analogy of how many angels can dance on the head of a pin, which was sort of this anti-science thing that was a debate in the church, but we're seeing that really that's what you're seeing in the roots of plants or inside of cells. It's just this amazing plethora of living things. I'm wondering, it's sort of similar to the virus question, but we're hearing a lot, and NIH has interest now in RNA communication. And I'm just wondering how much you've looked in these microbiomes at whether there's RNA being transmitted about between pathogens and then between pathogens and hosts.
So there are certain microRNAs that are conferred across taxa that apparently influence the gene expression of an alternate organism, and we see that present in the rhizosphere of poplar. There are also small secreted proteins that really aren't there for enzymatic function, but are signaling molecules that occur prior to physical contact. So the bacteria and fungi excrete a large amount of these very small 50 amino acid proteins that move cross membrane, enter the nucleus, and then change the regulation of gene transcription. So it's a very similar idea to what you're saying, is that lots of times the association occurs not just at the biochemical level, but at the transcript level, and that transcription can be influenced by these signaling molecules.
Margaret?
It's one of the things that Marty brought up in his talk, and Gerard touched on it quite a bit, and Hannah as well, is the idea that that the very beginnings of life are really important for establishing the balance in the microbiota. And to some extent, of course, you know, all three of you study vertebrates. And so I'm wondering from Angela and Gerald whether or not the microbiota of young Drosophila and young poplar are critical for the establishment of the appropriate microbiota and the appropriate immune system in later life. And in the case of Angela, it would be separating the influences, the innate immune system from the innate plus adaptive immune systems. And down in the plants, it would be an even different thing. So what are your thoughts on that?
We haven't investigated that in any detail, but there are clear impacts of the presence or absence of the microbiota and pathogen challenge on the larval form on the subsequent response of the adult. So there is some sort of communication across metamorphosis in the insect system. The gut bacteria are actually retained across that stage, and so it's an open question whether it is mediated by the microbiota, by host gene expression patterns, or some sort of interaction between the two. And I think that there's a tremendous opportunity to investigate that as a system where there is a very sharp transition from one very different morphological form to another as a way to investigating these sorts of developmental effects. There's an awful lot that needs to be done in this area. Yeah.
I showed a slide where we did plant trap experiments looking at pine and oak and poplar. And poplar and pine have been separated for about 350 million years, so they represent widely divergent evolutionary pathways. One's a gymnosperm and one's an angiosperm. And yet, when we did the plant trap experiments with azenic materials, we found that there was this core microbiome that could cross-infect or cross-associate with a broad array of taxa on the host side. So the current idea is that there's this core microbiome that's shared developmentally, that is recruited initially when plants are established in a new environment. That may, in fact, be common to root systems in general.
So if you raise the poplar for a certain amount of time in the absence of microbiota, is there any effect of that on its ability to interact with microbiota subsequently?
It doesn't appear to be true, not in the long run.
Okay, we're going to go to the microphone to my left. You've been very patient, Dr. Fishburne. Go ahead.
Thank you. A question for Gerald. So I couldn't help but to notice that there were rhizobium strains that were at least present, maybe selected in the endophyte compartment. As you know, the relationship between rhizobia and root nodules and legumes is a famous one, and they fix nitrogen in that context. I had thought, or at least I think the conventional wisdom is that relationship was restricted to legumes, but does your work suggest that it might be broader, that they're fixing nitrogen for poplar and other plants?
Yeah, that's exactly right. I mentioned on one of the earlier slides that poplar is a pioneering species and it invades open environments. And so the question was, how does this temperate species that represents one of the fastest growing tree species in the world achieve such high growth rates in a limited environment, or sandbars with limited nitrogen availability? Turns out that they do fix nitrogen. Nodulation isn't required for nitrogen fixation. And we just had a paper that's in press right now on that very topic. So there's a significant amount of nitrogen being fixed inside the root system, absence of nodulation.
Do you know if it's the same signaling system, nod factors and the like? Or?
It is not.
Dr. Hughes?
Hi. During Hannah's talk with the fasting during hibernation, my question is still ill-formed in my head, so please be patient. I was thinking from the microbial ecology perspective of a microbial community under stress and what kind of signaling a microbial community under stress would be doing, and then how that would interact with the human host or whichever organism, and how that could be a beneficial response as far as understanding what the ecological stress is that either the plant or the mammal or any type of organism is under. And I guess I haven't really heard a lot of research talking about the microbial community under stress signaling back and forth with the host, trying to adapt to the environment.
Right. I think a lot about this too, and my thoughts aren't fully formed either on it, but I think stress is relative, right? In this case, we have an example of animals that have been doing this for a long time, evolutionarily, along with their microbes that have been doing the same thing, and this could be an example of a community that is extremely resilient under stress conditions, and there may be levels of certain organisms that the individual organism adapts. I think back to that science paper, Justin Sonnenberg, Jeff Gordon, with B theta, where they showed so nicely that when they fasted the mice for a few days, and B theta changed its complement of enzymes to be able to forage on host mucins with that change. So we may have in the hibernator situation some species that are adapting themselves, and in other cases, the organism is the community of microbes is the organism that's adapting. So some are dropping down, going into some kind of a dormant or resting state to be seeded later in the spring, whereas others, like the Verrucomicrobia, that are very well adapted to continuing to forage in that very altered environment, become prominent. Now, the downstream metabolites that the Verrucomicrobia produce or other mucin degraders produce might be feeding those others that normally rely on dietary constituents, but yet they'll feed off of those species that are able to persist and live in that environment. So, I think it makes you think about how the superorganism of the microbiota or the microbiome as one organism is adapting to this, quote, stress, that of course it's very well adapted to, because this happens over and over. I often think that the first thing in a ground squirrel's life is that fetal transition, as it goes through the change in its microbiota upon weaning. And then every year of its life, the microbiota is undergoing a transitional period, and the immune system appears to be making that change on the host side with the animals too. So that's why this idea about to understand it all, we need to also think about what the host is doing for the microbes. So, for example, there's some evidence that microbes can induce in their hosts certain fucosylated glycans and causing the epithelial cells to produce substrates that the microbes can use. And we tried to start to take a look at that, if there are changes in glycan type on the enterocytes in the winter versus the summer, or mucin. We have some evidence that mucin genes are increased. There's increased expression in the winter versus the summer, perhaps to feed those microbes that the host is contributing, so that it's not as stressful for the microbes.
Microphone to the left.
Oh, Paul Fidel from LSU. I want to touch a little bit on the hibernation biology as well. It would seem to me that it would be incredibly interesting to find out the associations when hibernation goes wrong, and following up what Michael was saying, either naturally or forced, since you have the ability to manipulate. Are there microbiota changes that are associated when things go wrong? Is there anything known about that, or plans to do things like that?
No, there's nothing known. We're just trying to get a handle on what's happening in the normal state, although we are starting now to perturb the communities with antibiotic administration in a way really just to try to get it knocked down so we could ask some questions about the interactions. I can't tell you. Really, in hibernation biology, which is a century old probably, people have just not been thinking this way at all about the role of the symbiotic relationship and how that might be affecting. So we just don't know.
It would also seem that you could learn a lot about the factors associated, at least at the microbiota level associated with obesity or fasting, and all of the various issues that might be able to be exploited for treatments and so forth.
Yeah, it's a nice model in that way because we have both the obesity side in one part of the year, and then we have the absence of enteral nutrition and the effects of fasting on the other side of the year, and the animals are doing it beautifully. In fact, talk about stress, I will just say that when the times where our ground squirrels have not been allowed to hibernate because we don't put them in the cold room and really let their phenotype be expressed, that appears to be a stress situation for these animals. So undergoing this, what we think of as a stressful event, is something that's very much needed by them. And it may be the same for the microbiota, too, that this cycling is part of the microbiome's natural history.
Can I take off on that just a little bit, Dr. Carey? There is an example right now that's ongoing of a mammal species extinction that's linked apparently to a foreign organism that induces some change in hibernation. I'm talking about the bats, of course.
Geomyces.
Yeah, the geomyces. And I just wonder if you have some thoughts on how that's happening, because that's the clearest example of waking up the species completely. It's not just like it goes into a bit of hypermetabolic state for a while. It wakes up and flies around at night out in the snow.
Mm-hmm. No, that's a great example. We have kind of a hibernation-linked disease that has huge ecological implications because of the changes in bat populations. And actually, some of the work is being done in Madison at the National Wildlife Health Center, and we've interacted with those folks because so little has been known about immunology and hibernation at all. That there's now a resurgence, a real interest in thinking about the immune system when animals undergo these changes. So what we know, what I know of what's happening right now, and it very briefly is that, yes, this infection seems to be more prevalent. The animals are more susceptible than in North America than in Europe, where the organism, the fungus, has been identified before with bat populations. And the effect of the infection is seems to be causing more frequent arousal. It may have to do with the wings of bats are an extremely important part of their biology in terms of water loss and water balance. And these periodic arousals to euthermia, at least for very small hibernating species like a bat, may be linked to water balance changes. Could be happening in ground squirrels too, but I think especially for the bats. And this the microbes, the fungus is white nose, but it also gets on their wings. So these frequent arousals are causing their fat stores to be depleted at way rapid rates. And the response, and that's happened once in a while in our ground squirrels, when you get too lean, you just arouse and you start looking for food. And that's what's unfortunately happening, I think, to these bats. It's a real important issue.
So one of the things that occurs to me is that while humans don't hibernate, they do undergo a pretty profound day-night cycle. And recent data has shown that there's a circadian rhythm on the mucosal immune system at the level of the transcriptome, with all the clock genes involved in that. And also there's a circadian rhythm on the epithelial cells with all the clock genes cycling on a day-night cycle. And what's always occurred to me when I've read these things is there any effect, or how do we get it, whether or not the bacteria that are in the gut may be involved in setting the rhythms of the mucosal immune system and the epithelium, or be affected by them? Do you guys have any thoughts on this?
Well, raw gamma T, this nuclear receptor, which is driving this type 7, the cold immune response, is part of a family which is involved in circadian rhythms. So its closest cousin, raw gamma, is involved in the liver circadian rhythms somehow. And the other thing which is interesting is that raw gamma is a nuclear receptor, so it can theoretically bind some small ligands. It might be interesting to think of ligands produced by the microbiota, which would, in a circadian way, drive this raw gamma T or this containment response of the gut. There's absolutely no data to support this. Yes.
David, is there any push to look at, I don't know, maybe it's too complicated, the transcriptome on the day-night cycle in mammals at all?
In the microbiota, you're talking about? Yeah. I know there's been certainly talk and some thoughts about how you would design the experiment. It'd be certainly easier in an animal that is more amenable to manipulation and deconstruction, at whatever time of day. But I think it's a great question. You're competing with a number of other factors that have their own time cycle, and including not only just a variety of hormones, but changes in peristaltic activity, and how and where you would look at what would really be the key question. I still think it's a great idea to explore.
Yeah, even to just look at the transcriptome. I think it's interesting that bacteria seem to be involved in driving sleep cycles. And so, I don't know. I think it's...
Yeah, I agree. I had a question that takes us a little bit into a different direction, but Dennis Kasper has done some really interesting work, I think, showing that this deficiency in various immune system maturation phenotypes in a germ-free animal, although seemingly corrected after birth with restoration of its own native microbiota, is not the case when you give, say, a germ-free mouse the microbiota of a different host species, say human microbiota. The restoration of immune cell functions and numbers and distribution, all those sorts of things, is not at all, fully restored. And so it gets this idea of host species specificity. And even though that we're looking at what might be, really important phenotypes, we're doing it in a somewhat simple way, understandably. But when one looks in detail, perhaps there is specificity to each of these host species and their particular communities, and it would make sense, of course, that they've sort of co-adapted to each other over longer periods of time. Any of you have thoughts about what could be some of the mechanisms and bases that underlie this host species specificity to the kinds of interactions and phenotypes.
So the study you're mentioning is about one very particular bacteria, and this is the so-called segmented filamentous bacteria. It's about this, it's one of the few which can cross the mucus and bind directly to epithelial cells. And all the impact it has, and there's great impact on the adaptive immunity in terms of antibody production, cytokine production, and it binds to epithelial cells. And I think that's the answer to your question. Somehow the receptor which it binds to, or the exchange of metabolites it does with epithelial cells, because it's a very heterotrophic bacteria. It has a very small genome of 1.5 or 1.3 megabase. It does not cross species.
So I guess your answer is that there, even though there may be SFB equivalents in humans, which there almost certainly are, and all the other animal species, that the nature of their interaction, say, with receptors is the basis for the specificity.
That's what yes.
So, okay.
Yeah, we don't know the receptor, by the way. We don't know how it works.
Right. Right. Yeah, I would think that there might be also interactions or group effects that are also have specificity, besides some of these, direct one-on-one encounters. But it could be all of the above.
Well, I would think, like, things like short chain fatty acids, things like this have to cross borders, of course. But I think there should be such a tremendous effect on the immune system that it makes sense that it has a dominant impact on the immune system that does not cross species because of its very specific behavior. May I just add one thing to your point? You may know this paper from the 70s showing that people with sleeping disorder have this S factor in the urine, right? And this S factor turns out to be peptidoglycan and not two ligands. And in rat models, it has been shown that rats with sleeping disorders have PGN in the, I think, hypothalamus. So there is a link. There might be a link. We don't know if there's a causality on it, but there might be a link to sleep and getting rid.
Yeah, so is there a rhythm to the expression of receptors, or is there a rhythm to the presentation of peptidoglycan in the gut that then goes, of course, into the general circulation to the brain and then trains sleep cycle?
I'll tell you next year. We have to do that. And Ivo Boneca, with whom I'm working, has means to titrate the whole PGN production specific for node 1, node 2 in the microbiota. And we'll see if it's a winner.
I'm going to take one last question. Microphone.
Hi. I was wondering if any of you know that there are any differences in the epigenetic changes in the host that are induced by the microbes that could explain for some of the phenotypes, like the obesity phenotypes and others. Or if anyone's looking into the epigenetic changes induced by microbes.
I think there are examples in Shigella, but I'm not totally sure. Philippe Fontanet at Pasteur has published some papers on how Shigella does modify histones and responses to immune genes. That's what about I know. And maybe Listeria. There's another paper from Pascal Costa on Listeria, where she showed that it modifies histones.
Make one comment. Butyrate, when some of the short chain fatty acids are noted to affect histone acetylation, so it wouldn't be crazy.
Okay, that brings us to the end of the session. Thank you very much for great talks and discussion.